Значит, сегодня наш гость доктор Гieger, который сидит в первом ряду и готовится вам рассказать про атласы. Значит, я его сейчас представлю. По образованию он математик, статистик, и жизнь его связана в большой мере с Баварией, с Мюнхеном, где он посещал университет. Один известных баварских университетов, там их два: технический университет Мюнхена и Людвигс-Максимилиан университет, который, собственно, университет. Кристиан, он в университете учился, там выполнил свою дипломную работу в 1993 году, а в 1998 году защитил кандидатскую диссертацию, то, что PhD, на тему, которая весьма статистична, это анализ лонгитюдных исследований с использованием сплайнов. То есть это имеет отношение к эпидемиологии, но это имеет отношение к такой очень серьезной математической статистике тоже. Далее он несколько лет работал в индустрии, работал в таких компаниях, как IBM, в компании Lion Bioscience, которая была на тот момент одна из крупнейших европейских биоинформатических компаний. И после нескольких лет работы в индустрии он пришел обратно в академию. Сначала работал несколько лет в Фрауенхофен институте, а начиная с 2004 года, на протяжении уже 10 лет, его работа, жизнь связана с объединением Гельмгольца, а именно с центром, который находится, очень большой, серьезный центр в Мюнхене. И в этом институте он дорос до главы научно-исследовательского отдела, который имеет название молекулярной эпидемиологии. Мы с Кристианом сотрудничаем довольно давно. Мы оба, начиная с самого того момента, когда начались полногеномные анализы ассоциаций, исследования полногеномной ассоциации, сложных признаков, мы оба в этой теме поучаствовали. И поскольку в этой теме очень плотная связь между исследователями, на данный момент, наверное, у нас 15 или больше совместных публикаций. Первая датируется, наверное, к 2008. И вполне было естественно, когда объявили конкурс совместных программ РФФИ и Центра объединения Гельмгольца, что мы подали на эту программу и получили совместный грант, который продолжается уже 2,5 года. Вообще эта программа на 3 года, то есть в этом следующем году мы заканчиваем. Но мы очень плодотворно поработали по теме, которой Кристиан, собственно, и занимается. Это связь метаболома, генома и сложных заболеваний. Это, собственно, то, про что он будет говорить. Это представлять плоды многолетних усилий, трудов. Что еще хотелось бы упомянуть? В этом году был сделан рейтинг ученых в разных областях знания, сделан по цитируемости работ. И в этот рейтинг вошли те люди, которые в конкретных областях знания вошли в самый верхний процент, первый процент, 1, по цитируемости. И в области биологии и генетики Кристиан вошел в этот список, так что мы очень горды, рады за него и горды тем, что мы с таким замечательным большим человеком сотрудничаем. На этом я, наверное, закруглюсь и дам слово Кристиану. Кристиан, now floor is yours.
Yes, so thank you very much, Yuri, for this nice introduction. So I understand almost everything, but I have to say it's much more than we talked about before. So there must be some things which I not really can understand, but I hope it was, or I'm quite sure that it was a very nice introduction of myself. I'm really happy to come once again to Novosibirsk. So we have still our, as Yuri told you, our very good collaboration, and all. I like it very much to come to this part of the world, which is not, well, for me at least, the most usual part to go, but it is very nice, and I really like to come to Novosibirsk and talk with you and work on this collaboration. So thank you very much for this invitation, also for this invitation to hold the presentation here in your seminar series. So you can see my title of the talk. It's a quite general talk, but I hope I can convince you in the end of this talk that really are doing what we want to do, namely to do something which has to do with diseases and fight of diseases, all these things. So everybody knows, or everybody can see, people are different. So some of you are tall, some of the people are smaller, some of these people are slim, obviously, others are not so slim, and all these things. And for us, a very important question is, why is it so? So what makes us different in a certain kind? So some of the things are clear. I mean, people are eating different things. So people are either like this guy here or like the other guy here. Some of the people are making a lot of sports. So again, this guy here probably is one person who makes sports. This one there probably less sports and other things. This is kind of environmental factors, or for example, diseases can play such a similar role. And the other thing which is important is here this on the right hand side, where you see a DNA. So one reason that makes us different is different makeups in your DNA, difference in small pieces on your DNA, and all these things. So it's people getting more and more insight what's happening in humans. But one obviously very interesting thing is how these environmental factors, let's say, play together with DNA, with your familiar predisposition and all these things. And there's one thing which is really a major point where you can measure this thing where these environmental factors play together with the DNA and genetic things. And this is on the metabolome. So the metabolome, for those who do not know it, represents our metabolites in a biological cell or tissue. So in this sense, we can measure small molecules like vitamins, lipids, carbohydrates, and nucleotides, for example. And with these metabolites, you can really measure very exact what people did before, for example, in their life, or one can really measure very precisely the consequences of genetic predispositions and all these things. So the metabolome is, in a certain sense, the end product of the genetic setup of an organism, of a person, and it's the sum of all exposed influences on the organism, due to such things like nutrition, environmental factors, or drug treatment, for example. So it really makes a lot of sense to measure concentrations of these metabolites, or concentration of the whole metabolome. And that's what we are doing. So from a more general point of view, my group and people in Munich, for example, but other people here in Novosibirsk and another research institutes, is that people are looking not on diseases, but they look on something what we call intermediate phenotypes. So what is the intermediate phenotype for us and for others? It's a quantitative trait set or traits that are known risk factor for complex diseases. for example, things like blood pressure or lipid values in your organism. And it's assumed the case that these risk factors, quantitative traits, are modulated by common genetic variances. These things are more appropriate for things like we have in Munich. We have one large study, which is a quarter study. It's an epidemiological study, and this study allows you to measure this intermediate phenotype very good, because you can really measure it on population base, and it's not biased by certain diseases. But still, we are measuring not disease directly, but by looking at these intermediate phenotypes which are connected to certain diseases, we gain a lot in the direction of understanding disease pathogenesis. So this is exactly what we want to do with our group, with the data here, which we have in Munich, and where we also have found these results together with others in the next few slides. On the next few slides. Again, just to make it clear for one of you, so we have here our SNPs. So we measure in our studies on genetic side SNPs, and we measure in our core study, for example, environmental factors like how many food people are eating, or how many drugs they are eating, and how many sports they are making per week, and so on. And what we want to do in this study is bring both things together and try to find out if there is an effect on clinical phenotypes. And we do it in a way that we put in the middle of both extremes our intermediate phenotypes, measure this intermediate phenotypes, and draw conclusions on measurements of these known intermediate phenotypes. And in our case, we use as intermediate phenotypes metabolites. So now in the rest for the talk, I want to give you 2 or 3 examples. The first one is one where we're working since 7 or 8 on. It's an analysis of serum uric acid levels. So uric acid is probably one of the most famous biomarkers or intermediate phenotypes. So uric acid, it's known that complication of uric acid is gout, are kidney stones, and are urinary stones, but it's also known that there's a correlation, association to diabetes and cardiovascular diseases and all these things. So it's really a very typical and very useful biomarkers. And we looked at these metabolites. They are obviously relatively easy. 2 reasons for high uric acid levels, and high uric acid levels are the consequence, for example, for gout. So one reason could be the increased production of uric acid in the liver, and the second reason could be the insufficient uric acid excretion in the kidneys. The point was, 10 years ago or something like that, it was known that there is a strong genetic control that influences regulation of blood uric acid concentration, but there was no major common genetic variation known at that point. What was a little bit surprising, because there were a lot of studies on uric acid. What we did together with colleagues, we built up one of these genomic association studies. This means that we made a meta-analysis on uric acid, brought together 36 studies, and included in the end almost 150,000 participants. And after you do such a huge effort, you get something like that for this Manhattan plot. I hope you have some of these plots up to now. Here's are the chromosomes, here's minus log p-value, and each of these bars here is now a place where we got a significant results for a SNP which is associated with uric acid levels. And in the end, we ended up with 28 different loci where we could say, okay, there's an association between genetic loci and uric acid values in this case. So it's quite different from 10 years ago where almost nothing was known about the genetics of uric acid levels. In parallel, we did the same for gout. So we know that gout is a consequence of uric acid levels, and so we looked at the outcome of high uric acid levels. And what we got here is a Manhattan plot which looks really different to the last one, because for gout we found only two really significant loci for gout. And now, when we make a plot like this here, where we show the effect of increased uric acid levels and increased risk for gout, you at least see that there's an almost perfect association between these effects in uric acid and effect in effect on gout. So at least you see that we are looking at the right thing, but for uric acid we got significant results, and for gout we got only two genes which are increased the risk for getting gout. So this maybe makes it clear that it's more useful to look at intermediate phenotypes, because at least the statistic power is much higher than when you look on diseases itself. And this is a situation which you always, or in most of the cases, which you are always see when you look at intermediate phenotypes and outcomes or diseases which are correlated to these intermediate traits. So, as a summary of these projects which ended last year, so we found almost 30 genomically significant loci for uric acid. We found only 2 loci in gout. I already said uric acid increasing alleles are increasing risk of gout. We saw that the strongest loci harbor genes that code for renal transport proteins. This was also a surprise of us, because we really saw the most important place in the body is obviously is kidney for uric acid. Only those loci with smaller effects are coming from the liver, and within these genes which are coming from the liver, many of which may play an important role in metabolic control of uric acid and uric acid production and uric acid elimination. I will show on two slides where we really see that these analysis really makes sense also for somebody who is interested in the treatment of uric acid and gout. So, for example, this one is SRC2A9 protein was in our uric acid genome analysis additive and had a really large effect size, namely up to minus 0.4 milligrams per deciliter. So that's really strong. And they explain, or they've explained variances from 1.2 for men and 7% for women. And it could, followed by the first, she was shown that SSC2A9 functions indeed as a high capacity urate transporter. And we could show for gout that these loci has an odds ratio of 1.68. So it's also, at least for in these genetic studies, a really huge odds ratio which we observed in this for this gene or protein. And the same for our second strongest loci, which is ABCG2. Also here we could show that ABCG2 is uric acid transporter. Again, a new finding for biology at this locus. And we found that variant results in 53% reduced uric transport rates compared to wild type in mouse experiments. So you can imagine it's a really big effect when you have when you are able to, or when your transport is reduced by 53%. And again, also this variant or this loci shows a highly significant association with gout, again with 1.68 per risk allele. And for us, it was one of the largest meta-analysis on this genomic data. But this analysis really convinced us that these genome meta-association studies are really worth to look at, and they really deliver good results. And for us, these examples are really examples for the power of genome meta-association studies, in particular with metabolites like we do it. And obviously, these things somehow deliver an understanding of the disease mechanisms which were the unknown up to now. So this was our publication. And now, well, obviously, it's possible to do this analysis with one metabolite, like we did for uric acid. Others do it for glucose values and all these things. So it's not a problem. It's possible, but we thought from this point on that there must be something else, not just one metabolite after the other. And at the time, it would be possible to measure metabolites really as a panel of different metabolites. This can be done with typically with NMR experiments or with mass spec experiments, and you get a complete or more or less complete panel of metabolites in one run. And at that time, we decided to do this in our core study, in our large population-based studies, and try to analyze these results and try to find more interesting outcomes. So at that time, it was several years ago, 2009 or 2010, but at that time we were the first group who really could publish a series of genome-wide association studies with metabolomics. And at that point we saw already that we have similar results like in the analysis of uric acid. In some cases, the effects were even stronger. So we also got, for metabolomics analysis, large effect sizes, and we saw very often an overlap between our locus, which is relevant for metabolite, and so we got very often an overlap with risk loci of complex diseases. So the same locus is a locus for our metabolites and, for example, for diabetes or for cardiovascular diseases, like you could see in this series of publications. To show it a little bit more in detail, I will go in detail for two of our most recent studies. So the first one is again a genome-wide association studies with metabolites. We had two populations. So for uric acid, we had almost 60 studies, but for metabolomics, it's only there are only smaller sample sizes possible because not so many studies have metabolites available. In this case, we start with two studies. It's called F4, which is one of our studies where we have more or less 1,800 persons in the study, and this is K, which is a study which is a British study where we had at that time 1,000 participants. We used, as I already told you, one of these mass spec experiments, which is in our case an experiment or a setup which comes from a company in the US, from Metabulon. And in this study we were able to measure 250 different metabolites. So it's really a bit more than just one metabolite. And these 250 metabolites cover more or less 60 biological pathways. And what you see here is a cartoon of the results. So you see here, in different colors, we see different groups where we can find loci where a gene or protein or enzyme placed together with a metabolite. So, for example, this one you would already know it's uric acid and our FAC2A9, for example, or this is another one. It's FACS gene, which plays a role in certain lipid pathways, and so on. So we could go through all of these associations between genes and metabolites. So next thing what we did is, from experience in earlier experiments, we looked how is the overlap between the loci which we found in our experiment, in our metabolomics GWAS, and we found immediately that some of these, the yellow ones, have an overlap to associations with disease risk factors. So some of these associations were already known. For example, this FACS gene, for example, was already known as having an association with different lipid parameters, HDL, LDL, and so on. And the red select elements have already an association with a disease endpoint. For example, here CPS1 is associated with kidney diseases and so on. So we can also again immediately draw the conclusions that our results from metabolomics deliver either known associations, or for the others there's a chance that we could find also associations to other risk factors and diseases which are not already known here. And the same for overlaps to things which are known for correlations or associations to the function of drugs. So either that we find some loci which show genotype dependent responses to drug treatments, or we already know that there's a certain functional link to drug action in the other case. Again, our publication to this experiment, to this project. So we were really, at a certain point, we were really enthusiastic to got these nice results, and we decided, okay, to make our experiment larger. And the only difference here is that we have for the twin 6 times more people compared to the former experiment. It's the same metabolomics technology. There's one little difference, which means that we in the first project used only known metabolites. Known means that I really know what is metabolite which I saw in the mass spec. In our second project, we also used what we call unknown metabolites, meaning that we knew, or we know that in the mass spec I can observe a peak which must be a metabolite, but I do not know what is the name of the metabolite behind it. So we looked in this experiment at 513 known and unknown metabolites, again covering more or less 60 biological pathways, and in this experiment we finally found that we deal with more or less genomic significant associations at 145 metabolic loci. So we could increase by increasing the sample size of the UK twins from more or less 1,000 to 6,000. We could increase the found significant loci from more or less 40 to 149. And really, without going into details, what we see here is now a map of metabolites. Here, for example, as a circle where we know there is an association of these metabolites to all the genes, drawn with diamonds. So we know this metabolite here, number 35, has association to all these 8 metabolites around this genes around this metabolite. And now we think, and this is the reason why we call it Atlas, is the first thing that we really have a kind of map where people can navigate somehow through these network and found what are the metabolites which we found in our analysis and how they are also interconnected through to our analysis. And again, we did the same game again. So we looked there's an overlap to already known information about, for example, information about complex states with diseases or other outcomes, or to drug response, or if there is a kind of association with of drug targets or drug metabolizing enzymes or transporter proteins. And the third thing we looked up is that we looked at already known inborn errors of metabolism. And again, we get a lot of loci which are already known, and in the same sense, like in the last project, it really helps to put these results from our GWAS, put it somehow in the rest of the analysis in this field. Okay. This was our most recent publication, and in this publication we really found that it's worth to call it really an atlas of genetic influences on human blood metabolites. How much time do we have? Yeah. How much time do we have? Okay. And now I want to give you a little bit an introduction in our newest project or projects. So what we did up to now and what we will do also in the future is that we look at the genetic variances like SNPs and metabolites. But nowadays it's also possible not only SNPs but also it's possible to measure methylation sites on your DNA. And in the meantime, there's Illumina, one of these big companies. He was able to produce a chip technology which allows you to measure 450,000 of these methylation sites simultaneously. And what we did is that we used this technology and measure these chips at the same sample of our coral study, and looked what's the difference and what's the similarity to things which we got in our genome association studies. So again, one of these Manhattan plots. So we have in fact two Manhattan plots. One is a Manhattan plot which we get when we do this analysis for all 150,000 methylation sites here. And the second one, which is going to the bottom, is the same for an analysis where we adjusted for genetic variances in the same region. And what you can see here on the top panel here, again, you see we get a lot of very significant associations between CpG sites, so methylation sites, and metabolites, again. But some of them, or most of them, vanish when we adjust for genetics in the second analysis. So meaning that these methylation sites are very much associated to genetic variances which are in this area, which makes a lot of sense. But still, we have some findings, new loci, which are really new. So this is the first one. So we found a little, not many of these loci here are already known from genetics. So there's simply an overlap between methylation sites and genetics loci. And for some of them are really shows that there must be an overlap between genetics between SNPs and methylation sites, because for these four different genes, we could show that there's really a SNP within the CpG site. So it makes really sense. There must be an overlap of both effects. For some of them, we see that there must be a genetic influence for other CpG sites there, but we couldn't really see things directly from looking at our data set. So it could be, for example, that there are CpG sites which are correlated to our CpG site, which is significant, and this associated CpG site is driven by genetics or things like that. But we are sure that really the reason for these associations in CpG sites is in fact that we observe genetic variants at the same locus. So what we did in some analysis is that we put 3 SNPs from the same area in our epigenome-wide association study, and made the same experiment again, and by including our SNPs, which are in the region of the methylation site, we are able to correct the statistical site for these genetic influences, and what we get back is really CpG sites which are only driven by things like environmental factors or other things which can be the reason for differences in the methylation site. Again, a list of metabolites which we found. From this longer list, we could at least find or annotate 7 distinct loci. Here are the names, and they are really only associated with one metabolic trait, so it's relatively easy then to interpret these association between gene or protein and metabolic trait. And some of them are quite interesting. For example, this TXNIP is a gene where before groups have found an association between the gene expression and diabetes. In our case, the same locus was associated with metabolite which plays a role in diabetes. And the problem before was that the group which found the association with TXNIP couldn't find a reason for this variation in the gene. And now we think that the reason is not a SNP or it's not a genetic variation, but it's a variation in methylation patterns for this gene. The problem a little bit with these methylation or epigenetic project is that effects not comparable strong like in the genetic case. So we expect that we need more samples to get really more interesting results. And the other thing with these methylation patterns is very often that you do not know really what is the reason and what's the consequence. So it's not like in genetics, you know, as the genes are the reason and effects in metabolites is the effect. It's much more difficult for these methylation factors. For example, BMI is really a driver of changes in methylation patterns, and so you have a quite complicated interaction between relevant methylation patterns and metabolites which are closely connected with BMI, for example. Again, our publication, and well, these are my example where I wanted to show you how it works, and it really works, and you get really very nice results. And we really think that, something I already mentioned, that by going into this field of metabolomics and genetics and epigenetics, we can really help and deliver somehow an improved understanding of metabolic diseases. And we are quite sure that it's true, and we have already seen many of these examples. As a byproduct of these experiments project is that we really learn a lot about genes. So for many of them, of the genes where people not really know till now what is the biological function of these genes, these metabolomic experiments really help here to find it out. We have seen very often that there is an enrichment of genes, so it's an enrichment of genes which are already known to have medical and pharmacological importance. So we can really go to and take this information which comes from medicine and look what is the reason regarding the biological pathways which are affected. And in the end, we are quite convinced that these experiments are able to really deliver our part to find out what could be new pathways which underlie associations with complex diseases. So finally, I want to just mention some of the people. So I'm not really alone on things that most of these papers or projects are really big projects where people work together from different groups, from different countries, and so on. So here's just a list of some of the people who are working on this specific project, but this list is obviously not complete. There are other people who are working also on this different projects. So thank you very much for your attention, and as I said, hopefully I could show you a little bit that these are really projects where we think these things make sense for all of us. So thank you much.
Thank you very much. Christian, for this very interesting talk. For me, it was also very thought-provoking. Now we have time for questions.
Yeah.
Now it's possible to
Good minutes.
in America now you can pay $1,000 and get some of your SSDs sequences, and you get some information about your some SSDs that know to report some diseases. Do you think that right now, today, 2014, it makes some sense, or we should wait until, I don't know, several more years to know whether these SSDs are important or not? Is this information that you can get for this commercial information relevant or not?
Could you please repeat what do you mean is relevant?
I forgot the name of the film, but in America you can 23andMe,
Okay.
you can get DNA and they give you some information about your SNPs. Nowadays, does it make sense or not?
I think for scientists this makes perfectly sense because it's exactly what we're doing. So we sequence DNA and try to work with with this information. For me, a little bit the question is if this really makes sense for somebody who is not a scientist, because it's a bit hard, at least for me, that somebody predicts that I will get Alzheimer with 72 years and 20% chance. So what is the meaning in such a statement? And I think the problem is a little bit that you cannot deliver somebody who is by such information that you have a certain chance to get it. You have to find somehow a way to describe it to the non-scientists. And I think there might be somehow a problem because then the statements are so simple in a certain sense that it's maybe getting a little bit problematic, at least for me. But to coming to the beginning of the question, I really think that to sequence persons and analyze these data sets, it makes really perfectly sense. But for me, it's more a thing which should be done by scientists, not the people who are just discussing it at home at your kitchen table. But I know other people have another opinion, maybe. Really.
I actually quite agree that, for scientists it makes sense that as for these direct-to-customer things, the results are mostly not very clear-cut. It's increased risks, and the question of interpretation, when you give to non-specialists this bunch of numbers, it's like, yeah, what's the value? So I think we quite agree on this.
Yeah. And in fact, we are really doing something like that. There's a database of 23andMe, and some sense larger compared to our database, but we're making completely the same. So we plan to sequence, for example, our samples from Quora, our own study, and so on. So I'm quite sure, I'm convinced that in some years, each of these persons in these big studies are sequenced. So.
Thank you again for interesting talk. My first question concerns the metabolome as a term. So initially you told that there is differences between two persons, but it's also known that there is also difference in metabolite levels for different cell types. And I'm not sure that it's a general situation, but probably for some complex diseases, the different cell types involved in disease development. So when we talk about the what is the metabolome? Is there some intermediate, as you said, intermediate phenotype, or what is it?
Well, first of all, I wasn't very clear in that point, because what we are doing in our projects, we have metabolites coming from serum. So, and it's quite clear that we do not measure every time the right thing, because for some metabolites it would be, or for some situations it would be much better to have metabolites coming directly from liver, or coming directly from kidney, or even somewhere in brain and all these things. But we are a study where this is not possible for us, because we are one of these large population-based studies, and the only thing you really get is blood, and sometimes a little bit fat, or a little bit skin, or something like that. So you will never get from such of these last studies biopsies of liver. So for us, because such things you can only do in hospitals. But you are completely right. For sure, there is a difference when we compare different tissues. And but we made the experiment that blood is not the worst tissue, because for us at least it seems a little bit this is somehow a kind of collection of everything. So a collection of liver, a collection of kidney metabolites, and so on. So in the sense, for us at least, we do the best in this area. But you're right, it is really a valid question or remark. Yeah. But I mean, maybe one thing. So we are not the only persons working in this area. For example, in Munich at the Hammer Center, we have a big groups which are working with mice. And in these experiments, it's possible to look at liver and kidneys and so on. So, and what we are really trying to do is not always so easy, but what we always try to do is to work together with mice people and mouse people, and somehow build projects together. So, for example, in a way that we are saying, look, we have found something in the humans. Could you do a little bit more on a special designed mouse experiment?
Are there any tools or approaches that provides opportunities to reconstruct the network or ways by which this genetic variations can lead to really metabolite changes? I mean, computer tools.
So you mean, can you?
Are there any computer tools that allows to reconstruct the network, not associated network, or ways by which this genetic variation can lead to metabolite changes to metabolite pathway?
Yes. So I have to admit that I'm not really an expert in this field, because we really thought that our strength is that we can build up these association structures only with our own data. So for us, it was not the goal to incorporate somehow all different databases. But there are tools, and but to be honest, I'm really not an expert in this field. So what we are doing is, what we try is to build up these networks which coming from this association analysis, and then we go into the database and look if there's, for example, something in this cat database, find out if we are able afterwards to annotate these metabolites. But what we not do is to build up these networks with the use of these software.
Other questions? Well, if not, maybe I could ask a question. And I like all parts of your presentation, but the gout story is quite terrific. Right?
Yeah.
And then I remember from your presentation there are these two loci with biggest effect, solid carrier something, SLC, and ABCG2, and they not only demonstrate its effect on the uric levels, but also are even is not so big gout. They have like not very trivial odds ratios, right? It's quite big odds ratios for complex disease. And then you also did functional experiments showing that they lose part of function, right? And then the question is, do you see, maybe it's speculation, could you speculate if these particular findings could have impact on personalized medicine?
I'm quite sure, because I know that there isn't, so I remember it extremely well, but for example, for the first one, for the SLC2A9, so people tried to build up a drug, a medicament, which is specific for the different phenotypes. But I'm quite sure, because it's obviously when you are looking for a common disease like gout, the first thing you do look at, and you want to build up a kind of personalized system to treat these disease. I would really start with this. This is the first and most influenced protein or gene. Yeah, I'm quite sure. And but I really cannot say it in detail. There are some investigations where people looked. I also think in mouse experiments what's happening under different conditions for mouse which have one or the other variant. No, but I mean, you know it as well as I do. Usually, you never get such a large effect for these genetic influences. Usually, you have unexplained variances of less than 1%, and in this case, at least for women, you have 6% explained variances. So it's not really the usual result. That's the reason why many of these people now starting to build up, or one of the reasons why many of these people now start to build up kinds of interaction models where I put together genetic information with methylation sites and metabolomics, and put everything together and hope in the end we will find something which shows really a clear result.